Hi, this is Mr. Evans. Welcome to another episode of Room 310 Biology. Today we're going to be focusing on membranes and what they're made of and transport. In other words, how materials actually are able to move in and out of a cell by getting through the cell membrane. The first question we might want to consider is how do you actually build a membrane? What are you going to build this part of the cell out of? Cells are mostly water, so we need a material that doesn't mix well with water. And you might be recalling from your chemistry section that the best material for not mixing with water happens to be a lipid. And sure enough, phospholipids are going to come to the rescue. Phospholipids are made of a phosphate head, which is a PO4 minus. We'll go ahead and write a phosphate head. This part of the molecule is actually what we term hydrophilic. It likes to be in water. It has an affinity for water. The other portion of the molecule are fatty acid tails, and the fatty acid tails have a slightly different property because of their being a hydrocarbon nature. They are hydrophobic. They have a fear of, not literally, but they don't have an attraction towards water. So cell membranes form a bilayer. That's an important term to realize. A lipid bilayer. The heads have an attraction towards water, and they face both into and inside the cell and away from the cell. And our fatty acid tails face one another, and this is the hydrophobic portion of the membrane, and it forms a wonderful barrier that cells will be able to make use of. So think to yourself, what kinds of things need to get into or out of a cell? Certainly carbon dioxide is a waste product that's going to need to move out. Oxygen is something that's going to be needing to move in. Amino acids is sort of raw products. Fuel source such as glucose, and without a doubt, water is certainly something that's going to need to move in and out, all in order to maintain homeostasis for the cells. Lots of molecules can move into or out of the cell without too much difficulty if they're small or if they are non-polar molecules, but some molecules will need a little bit of help. They have a lot of trouble getting through this barrier on their own, and because of that, there are many protein channels built into membranes, which may be specific for certain types of molecules, and maybe something needs to move into the cell through this protein channel here. There'll be a slightly different environment on the interior of this, so some molecules that can't pass through normally will take this route instead, and that would be called facilitated diffusion. So why do molecules move through? In the molecular world, molecules tend to want to move from areas of high concentration to low. Right now you see that this side of a membrane is highly concentrated with molecules of dye, and this side has none, so we'd say it has a very, very low concentration. As time passes, molecules will move across from areas of high concentration to areas of low concentration, and eventually are going to reach a point which we call equilibrium. Another word for equilibrium might be balance. But something important to remember is molecules will continue to move back and forth across that membrane in both directions. They just happen to maintain an equal concentration on both sides at this point at equilibrium. So let's see if we can take a look at this little graphic here and figure out which way these fat molecules would want to move. Fat is nonpolar, it's hydrophobic, it would have a very easy time moving across the membrane. There are more molecules of fat on this side of the membrane than on this side, so as you might rightly expect, molecules would move this direction until they can equal out a concentration on both sides of the membrane. Now, water gets its own special case of diffusion. Diffusion is a passive process. It doesn't require any energy input from the cell, but osmosis is the diffusion of water only. From areas of high concentration to low concentration. Down here we have what's called a U-tube, and this is not where you watch silly videos or watch my videos, but instead we have a glass tube. It has a selectively permeable membrane in between, and what we're hoping for is to see that water is going to move this direction. There are lots of solutes on this side, very few on this side. The water actually is going to move from an area of higher concentration of water over to sort of dilute all of these solutes. And you can see actually the water level rose on this side of the tube as water moved over onto this side. In certain cases, we see things happening to cells because of the environment that they are in and the amount of what's called free water. Here we have what's called a hypotonic situation in this first column. The concentration of free water is very, very high. So water is going to want to move into the cells, and because animal cells lack a cell wall, the cell can actually burst or lyse. Plants like a hypotonic situation. Water will continue to move in. Your vacuole swells up larger and larger and starts to actually press against the cell wall in every direction. Helps that plant stay upright. Very, very good situation for a plant to be in as a hypotonic environment. Here in the middle we have what's called an isotonic situation. Animal cells prefer this. Water moves in and water moves out at basically equal rates. Plants don't like this one quite so much. The vacuole is smaller here than it was in the situation over here, so your plant may start to lose some of its shape. You see the cell wall is not being pushed against as hard. The plant might look like it's starting to wilt just a little bit. And finally, our last column, no cells particularly like a hypertonic environment. There would be so many solutes dissolved outside the cell that water will want to leave both situations, both plant and animal. You can see that the animal cell will start to shrivel up. It's lost so much of its water. Same with the plant cell. It keeps its shape just a little bit, but because of the cell wall, but the entire interior actually shrinks up. And it's all due to the fact that water wants to leave and go to where the concentration is lower on the outside. A few organisms have special ways of adapting and handling these situations. This paramecium has a special organelle right here. This would be called its contractile vacuole. And the contractile vacuole functions to pump water back out of the cell. If too much water wants to move in, this organism lives in fresh water, it can actually use its vacuole to push that water right back out and thus maintain its homeostasis. So bottom line, what we would like you to take away from this quick lecture: membranes are made of phospholipids. It's a phospholipid bilayer. The heads are hydrophilic, the tails are hydrophobic. Membranes are selectively permeable. They are choosy about what can pass in and what can pass out of them. And finally, the idea of diffusion, that molecules move from areas of high concentration to areas of lower concentration. If those molecules happen to be that of water, we'll stop calling it diffusion and we'll instead use the term osmosis. So just a quick introduction to cell transport and membrane structure, and we'll see you again in room 310.